Good morning, everyone. Before we start our webinar, may I know, can everyone hear my voice? If you cannot, please leave a message in the chat box and let us know. So first of all, welcome to our webinar, CRISPR-transfection Efficient Genome Editing Using Nucleofector Technology, brought to you by NextGene Scientific in partnership with Lonza. I'm Meng Chee, and I'm the moderator of this webinar session. Before we move on to the sharing today, I would like to seek everyone's cooperation to put your mic on mute. This presentation would last for about 40 minutes, followed by a Q&A session. If you prefer, you may drop your question in the chat box or the Q&A widget available on the screen. We would be happy to answer your questions at the end of this sharing session. Now, I would like to introduce our speaker today, Mr. Tung Chee Hong. Chee Hong is the Field Application Manager at NextGene Scientific with more than 13 years of experience with various overseas training from our principals in US, Canada, China, and Singapore. In the course of his career, he also has attended CRISPR training programs organized by Lonza and NEB using the Nucleofection technology. Besides that, he has been invited as speaker for various seminars and workshops to talk about the molecular techniques, cell isolation, stem cell, chromatography, and many more. Without any further ado, I would like to welcome our speaker, Chee Hong, who will take us to explore the world of CRISPR and Nucleofector technology. Chee Hong, the floor is yours now.
Okay, thank you, Mengti, for the kind introduction, and thank you everyone for joining the webinar today. Let me do a little bit setup first. Okay. Yes. Okay. Yeah. Okay. Can everyone see my screen now? Mengti, can you see my screen?
Nope.
Nope. What about now? So not seeing my screen? Oh, sorry, I haven't shared this. Yeah. Okay.
Yep, I can see your screen now.
Okay, one moment. Okay. What about now?
Yes, we can see the screen.
You can see my next slide also?
Yes.
Okay. Okay, thank you everyone for joining. My name is Chee Hong. Thank you very much for joining us today. Today I'll be sharing on a topic called CRISPR-transfection efficient genome editing using Nucleofector technologies. Hope that and all of us are able to learn something on today's presentation. Today, the sharing will be breakdown into four agenda. The first one will be a brief introduction to the genome editing. Then we shall proceed to genome editing tools with some brief overview. And then we shall continue with using the Nucleofector technologies for CRISPR-based genome editing. And finally, we will take a look at some tips and tricks for efficient delivery into primary cell and cell lines using Nucleofector technologies or other technology. Okay, so without further ado, let's start with the first agenda of today, which is a brief introduction to genome editing. Okay, so there are actually various options to modulate gene expression, and then this can be done on either on the DNA or RNA or protein level. Many of these options actually will result in a transient modulation only. In order to have a permanent effect, a stable heritable DNA modification was accomplished either by random integration of plasmid, transposomes or viruses, or even via homologous recombination. Okay. So the DNA modification resulted in a site-specific integration, but it's a very time-consuming and inefficient process. However, with the introduction of advanced genome editing tools, this site-specific stable modification can now be performed, I would say, more easily. Here, I would like to share some of the genome editing tools available. Okay, so the genome editing tools that we have currently is from what we call zinc finger nucleases, and the short form we call it ZFN. Then we have the transcription activator like effector nucleases, the Talent technologies, and these two techniques were established over the last decade as useful tools for site-specific genomic modification. They are not something new, but they do not gain much attention until recently. This is with the recent discovery of CRISPR, gene editing tools is now in the limelight, okay, because of the advantages that it can offer. Okay, so what is special about this CRISPR technology is that it is able to provide a targeted modification of a cell's DNA via genome editing, and is actually provide powerful tools for fundamental biological research, early stage drug discovery, and for the development of novel cell therapy. So for example, immunotherapy. Okay, so genome editing tools make use of engineered nuclease to delete, insert, or replace a gene at targeted genomic location. Okay, so the nucleases is actually generating the double-stranded break in the genomic DNA, and thus inducing one of the two possible cellular repair mechanism. Okay, so the first one will be what we call a non-homologous end joining, NHEJ, that will lead to a deletion of the functional gene, since it's often accompanied with mutation when closing the break during the repair. Okay, if a partially homologous donor sequence is available, an insertion or replacement of a gene can take place via homology-dependent repair. To perform such modification at a specific target sequence, the nuclease is fused or to interact with sequence-specific DNA binding component, directing the nuclease to the targeted sequence. Okay. Here, I would like to show a comparison among the three members of the genome editing tools. Okay, as you can see here, the DNA binding molecule of CRISPR technology is RNA. So let me open my pointer first. Okay, a pointer option. Okay, so you can see that in CRISPR, we're using a RNA DNA as a DNA binding molecule, whereas for the other two techniques, we're using a protein. Okay, so the difference is that this makes CRISPR easy to modify because we can just modify the RNA for a new targeting site. So it's easier to design because we know that RNA and DNA just need complementary of the usual one, A to T or A to U or C and G. Well, the protein binding to DNA is actually harder to predict. Okay. So multiple target, as you can see here, is also possible and becomes simpler. Okay, the only downside of CRISPR technology is that because it has only it's only targeting one 18 to 28 sequence. Okay, so therefore it has lower specificity. Whereas for ZFN and Talent, they are actually binding to two parts, 2x15 or 2x13. So they actually has higher specificity. And then they also have lower off-target effect. So because of the advantages mentioned earlier, because of the ease of use, CRISPR has become the obvious choice for genome editing. Okay, so the CRISPR technology is actually based on a bacterial defense pathway against viral invasion. It has been adapted for genome editing application in eukaryotic and many other cell types. Okay, it's an emerging technology because it allows for more flexibility. In contrast to ZFN and Talent, CRISPR-based genome editing is relying on two separate components working together. Okay, so the first part that is required is actually the DNA targeting part, and it's the part that we need to modify if we have a new target. So this part we actually call it a guide RNA. So in short form, we actually call it gRNA. So it can be designed and then complementary to a 18 to 21 base pair stretch of targeting part of the genomic DNA. So then the gRNA, we actually recruit a protein so called Cas9. So Cas9 protein is actually a nuclease that we actually cut like a scissor, cut the DNA. So you actually, these two will form a complex and then go to the targeted region of the genomic DNA and then introduce a double-stranded break at the defined site. Okay, since the DNA specific part is the RNA molecule, again, as mentioned earlier, it's easier to design and produce. And whereas ZFN or TALEN, we actually have to predict the protein binding to DNA and then also to synthesize the protein. Okay, so in addition, by transferring Cas9 nucleases together with several guide RNA, we can actually perform multiple genome editing at the same time. Okay, so one of the crucial steps in the success of a CRISPR experiment is to deliver the gene editing element into the living cell. So as mentioned earlier, the guide RNA and also the Cas9 protein. For this reason, the choice of transfection method used is actually of utmost importance. So here I would like to introduce nucleofection, which is actually a proprietary transfection method offered by Lonza. Okay, it's a transfection technology that give a high transfection efficiency combined with low mortality. Sorry. And then in addition, nucleofection has a unique feature in which the DNA is directed into the nucleus straight away, giving faster gene expression. So compared to conventional method, the DNA or the RNA or protein that transfected, we actually in the cytoplasm of the cell, so outside. Whereas for nucleofection, we actually straight away deliver into the nucleus, which has a faster result. So here I would like to share in-house testing from Lonza to showcase its ability to deliver DNA straight into the nucleus. So I hope that you can able to see the picture clearly here. So in this in-house testing, so we're using a plasmid DNA actually labeled with this frozen dye. Okay, so therefore the plasmid DNA is actually red color. And then also inside the plasmid DNA, we are coding a green frozen protein. So once the plasmid DNA is inside, you produce green frozen protein in a living cell. Okay, then after that, we can actually use DAPI dye to stain the nucleus DNA. Okay, so when we overlapping the three of this dye, we are showing that the plasmid DNA is inside the nucleus, and then expression of green frozen protein is actually happening. So nucleofection technology is actually a complete package. So it's combining the use of an instrument called Nucleofector, and then you actually offer, it need to be used together with a Nucleofector kit, and then finally it actually has an optimized protocol. Okay. So it's a unique combination of device, a kit, and a protocol. And with the combination of these three, is enabling excellent transfection performance combined with high functionality. Okay, here I would like to show to you the platform available. So for a throughput of 16, we actually has an X unit. So therefore, you can actually transfect suspension cell. So you can actually do passaging to a detached or adherent cell. And then you can at any one time transfect 16 different substrate, okay, in a single transfection. If more is needed, then we have also 96 and 3x6. And the same protocol can be actually transferred to a large volume nuclear transfection in case you need to transfect more number of cells, more than 100 microliter or more than 10 to the power of 7. So it's a seamless transfer of the protocol to a unit that we call LV. So nucleofection is actually pretty simple. So first we need to harvest cell by using normal passaging. Then we mix the transfection reagent together with cell, then DNA, and then we move it into the kit. The kit will provide a QVAC, And then after that, we actually transfer into the machine. And then within seconds, the cell will be transfected. And then we just need to culture the cell and waiting for the result. So because the substrate will be straight away delivered to the nucleus, so we only need approximately 3 to 8 hours to see the expression to happen. Okay, so currently there are more than 80 primary cell type actually successfully transfected, and also a lot of hard to transfect cells are able to be transfected using Nucleofector as well. Okay, so now we take a look at why should we use Nucleofection for CRISPR. Okay, so firstly, it has been shown to transfect many types of cells as shown earlier. Okay, so with this one, we have optimized protocol for many cell types, and that makes the transfection protocol low optimization effort. Okay. Secondly, it has high transfection efficiency for a broad range of cell types, including the very hard to transfect iPSC cell, or it also able to offer transient or stable transfection to hard to transfect cell line as well. For example, blood cell line and primary cell like blood cell, stem cell, neurons, and all that. Okay, so Nucleofection also offer efficient core transfection of various substrate as well. Okay, so because CRISPR experiments, sometimes we need to core transfect, meaning that we need to transfect not only plasmid, we need to transfect actually a combination of plasmid and DNA. So in addition, we are also able to transfect DNA, RNA, or protein. Okay, so here some condition for core transfection. So sometimes we need to transfect DNA vectors together with, let's say, the SSODN, which is actually for HDR targeted specific modification. We'll look at that later. Okay, so finally, Nucleofection is able to offer high reproducibility. Okay, so this slide is actually showing how the various type of genome editing using CRISPR. Okay, so for CRISPR transfection, there are several possible form of transfection. Okay, so I will actually categorize it into 3 types. So you can choose only one type. Normally, we only choose only one type. So the traditional or most, the first generation one, we actually transfer plasmid DNA. So I call it to use DNA. So the DNA will actually contains the Cas9 protein coding sequence, the gene, Cas9 gene, and also the guide RNA in the DNA sequence. So we have to transfect the Cas9 and guide RNA plasmid into cell and ask the cell and rely on the cell to actually express the Cas9 protein as well as the RNA molecule of the guide RNA. Okay, so sometimes we actually use 2 plasmid to code for the Cas9 protein and the other one to code for the guide RNA. Okay, so we will need core transfection of this. Okay, another one is when we want to do specific targeted modification. So we need to core transfect a plasmid carrying Cas9, and then after that we need to have another DNA element, be it a plasmid or a double-stranded DNA that we call a SSODN. It's actually a single-stranded oligodeoxynucleotide. Okay, so this one we actually do the specific modification. And finally, we have the third version that we call RNP. So we actually generate the guide RNA in RNA molecule form before we transfer into a cell. Okay, and also we actually has the Cas9 protein ready. So when we transfer, we actually transfer the RNA molecule with the Cas9 protein that form a complex, and then only we actually transfer them into a cell. So basically, cell no need to go through the events of expression to produce guide RNA and also Cas9 protein. Okay, so when aiming for insertion or replacement, okay, so most of the time you actually use the single-strand oligonucleotide that I mentioned earlier. Okay, so therefore it will actually require core transfection. Okay, here I would like to show a few experiments that using Nucleofection for a CRISPR experiment. Okay, so the group of scientists that is actually Young and co-worker actually published a publication called Optimization of Scarlet's Human Stem Cell Genome Editing, okay, back in 2013. Okay, they are using Nucleofector to actually transfect human induced pluripotent stem cell. Okay, so the main thing about this particular study is that it's comparing genome editing efficiency of TALEN versus Cas9 system. Okay, so both of them will be used to target chemokine receptor CCR5. Okay, so they actually want to have a specific modification. Therefore, they also co-transfect with the SSODN, the oligodeoxynucleotide. Okay, so as a result, we are highlighting that the repair mechanism NHEJ for TALEN actually achieve efficiency of 0.4% only, but when we're using the CRISPR system, 3% of the population of the cell actually detected with the NHEJ mutation. Okay, and then for HDR, okay, the modification using the SS oligo-DNA deoxynucleotide that has specific intended modification, TALEN only achieved 0.6% of modification, whereas the efficiency has been increased to 1% for CRISPR. Okay, so this particular publication showed that both nuclease platforms serve as a robust tool for genome editing, but the result from CRISPR system is actually achieve higher efficiency, which is actually approximately 7 to 8 times higher than in terms of NHEJ compared to TALEN method. Okay, another tool that experiment that I will highlight is actually the publication by Kim and co-worker. So for this particular publication, what I would like to highlight is actually the advantage of using the RNP transfection method instead of the plasmid DNA and or the mRNA method. Okay, so by using the RNP method, deliver of guide RNA complex with the Cas9 protein in a tube before transfection, it achieves mutation frequency comparable with plasmid DNA. But in another cell line, the mutation frequency is actually higher for fibroblast, the embryonic stem cell. Okay, so in addition to that, they also show that it has better viability after transfection when using ESC compared to plasmid DNA transfection. And most importantly, they show that the RNP method actually has reduced off-target effects. Okay, so in another publication by Lin and co-workers, okay, they actually show that they want to increase the efficiency of HDR modification, means that targeted modification. So what they do is actually they arrest the cell cycle and then after that combine with the use of RNP transfection, then they're able to get enhanced HDR ratio for HEK293 cell, fibroblast cell, and also ESC cell. Okay, so therefore they actually recommend the use of the RNP, and also this is because they enable the control nuclease action, and also together with the cell cycle arrest, they give a better result for the HDR targeted modification. And again, by using this, they are able to reduce off-target effect as well. Okay, so finally, the last agenda of today will be some tips and tricks for nucleofection and CRISPR experiment. Okay, as a general rule of thumb, okay, the cell has to be handled very carefully because they are very delicate, and transfection is actually a very slightly harsh step. Therefore, we need to make sure that the cell is happy before we're going through the transfection. So first thing is, of course, we need to count the cell, okay, using the correct cell number. And secondly, do not spin the cell too fast as it will cause harm to the cell. And then during the handling of the cell, pipetting also will have to be gentle, especially after nucleofection since they are like injured. So to minimize shear stress. Okay. And finally, after transfection, plate cell at a density that makes scientific sense to your experiment. If you're using optimized protocol from Lonza, you can actually get a recommended density as well. Okay, so how do you get an optimized protocol is actually depends on whether it's available from the Lonza website, or you can actually reach out to us and then we will get a recommendation to you. So in the case of there are no optimized condition, we also are offering an optimization kit to look for the best transfection condition to get the good efficiency and as well as a good viability using a control, a plasmid control called Pmax GFP. Okay, so before I before I continue with the tips and tricks, so as mentioned earlier, there are 3 possible delivery for CRISPR experiment. The first one will be delivery of the DNA, using the DNA for the CRISPR experiment. Here we are actually using a plasmid coding for guide RNA and also coding for Cas9. Okay. Secondly, we can choose a second approach, which is actually deliver in vitro transcribed RNA molecule or chemically synthesized RNA, guide RNA molecule, and also again in vitro transcribed Cas9 mRNA. And lastly, more recently, the most popular choice will be using guide RNA, which is an RNA molecule, and then also Cas9 protein. Okay, so if you are using the traditional method, which is actually using the plasmid DNA, okay, the quality of the plasmid DNA is very important. First thing will be the purity of the plasmid DNA used. So make sure that you use highly purified DNA. Okay, check the A260 and 280 ratio. So ideally 1.8 to 2.0, but 1.6 is acceptable. And another thing to highlight will be to use the endotoxin-free plasmid extraction kit when dealing with the mammalian cell. Okay, so to prevent them from having some viability issue. Okay, another thing quality check that we can perform is actually just to run gel of the plasmid DNA. So to check for nicked or degraded plasmid DNA on agarose gel. And the third one that we would like to check will be regarding the backbone, the choice of plasmid for the expression. So especially for the choice of the promoter. Okay, so plasmid DNA quality is actually very important. So it has been shown that nicked DNA may lead to low transfection efficiency. Okay, so normally we see the different conformation for plasmid DNA, whether it's open, circular, or supercoil. Okay, so you can see that the expression of a gene inside the plasmid will be affected if, let's say, the plasmid quality is not good enough. So we can see there are some expression level different. Okay, so and then the second one will be the choice of the promoter. So expression level of protein, for example, guide RNA or the Cas9 will be promoter dependent. Okay, so depending on the cell type, the promoter used can have a very different effect. So here we are showing a comparison between the SV40 promoter and also the CMV promoter. So you can see that CMV promoter works well for the THP1 cell, mouse T cell, and also HUVEC. But SV40 actually perform not very good in HUVEC cell. Okay, so therefore maybe some literature search need to be done before using the promoter of choice. Okay. Okay, so next one will be the co-transfection amount. Okay, so when using co-transfection, either when you are using 2 plasmid that code for Cas9 and guide RNA respectively, or you are actually co-transfecting with SSODN, single-stranded oligodeoxynucleotide, okay, for targeted modification purpose. Okay, So you might need to optimize the total amount of DNA and also the ratio between the different type of substrate. Okay, so here I would like to show an example range from the publication. Okay, so for Cas9 plasmid, the concentration used will be between 0.02 to 0.05 microgram per microliter. Then guide RNA also similar range. And then if you are using a donor, then the range will be slightly more, 0.02 up to the level of 0.1. Okay, so then after the ratio, we'll look at the ratio later. Okay, so if let's say you choose not to use the DNA method, you don't want to transfer the plasmid DNA into the cell, you might want to, you can also choose to transfer mRNA of the Cas9 protein. Okay, so however, when you you are transfecting mRNA, you need to know the nature of the mRNA inside a eukaryotic cell. Okay, so for eukaryotic cell, the RNA need to be to have some modification. So the first thing is you need to be capped, and also the second thing is need to be polyadenylated. Sorry about that. Somehow I was silenced. Minty, did you silence me? So sorry. Can you hear me now?
Yes.
Yeah. So I was silenced. Okay. Okay. So the mRNA should be capped and polyadenylated, and the optimal amount has to be titrate. Okay, might be higher than for plasmid. So it might require 2 to 20 microgram in 100 microreaction. Okay. However, even though high amount is required, the volume should not exceed 10% of the total sample volume. Okay. So depending on the cell type, for example, dendritic cell, it might be best to add the mRNA directly into empty cuvette and then add the cell solution mix on top. After adding cell suspension, then transfect the sample immediately. So the sequence of the addition of a substrate might be crucial. Okay, so further tips can be referred to the publication below. So Handel et al. At Nature Biotech, and then Bach et al. At Cell Report. Okay, so the third one will be when we choose to use the RNP complexes. Okay, so there are several advantages of using RNP complexes compared to the mRNA method as well as the plasmid DNA method. Okay, so first thing is no cloning of the construct required. Okay, secondly, the complex is actually less toxic than DNA. Thirdly, it's actually more stable than mRNA because, of course, RNA molecule is actually very fragile and easy degraded. And lastly, second, one advantage point before the last one will be no transcriptional process needed. Okay, so it's easier to control the dosage. We won't overexpress and have when we have overexpression using plasmid DNA, so it might actually bring undesired effect like the off-target effect. So define action time, so lower risk of off-target effect when we compare to plasmid DNA. Okay, so here I would like to show a study by Seki and Ratz, okay, in 2018. Okay, so this study is actually using nucleofection of RNP into resting human CD4 T cells and activated mouse CD8 T cells. Okay, so what we can learn from this publication is how they optimize a CRISPR experiment. In this experiment, they are actually looking into various factors affecting the success of a CRISPR experiment. Okay, So the things that they're actually looking into will be the effect of different nucleofection conditions to get the best transfection efficiency and as far as the viability, to determine the knockout efficiency by using different guide RNA, try to optimize the best Cas9 and versus guide RNA ratio. Okay. Finally, they're somehow showing that they could be different when we're using different Cas9, commercial Cas9 protein. Okay, so for your information, so Nucleofection is actually offering different transfection program. So for different cell type, we're actually offering different optimized protocol. So you can see that there are different code here. Again, I need to have my pointer back. Okay, so the EH100, DS150, and all that are the different program. Okay, so the program different. Hello. Yeah. Okay. So yeah. So you can see that there are different program here. Okay. So the program different in terms of the intensity of electrical pulse as well as the duration of the pulse. Okay. So in general, if the pulse apply is too strong or for too long, then the viability of the cell will be affected. Okay. However, if the pulse is too weak or too short, then the transfection efficiency will be low. So you can see that the result of here, in general, the transfection efficiency is good, more than 80%. But some of them actually for this particular program, the transfection efficiency and also the knockout efficiency is only 60%. So there will be a different when different program is used. Okay, so here are the same experiment by Seki showing that when we are using different guide RNA, it might have different effect. So in their study, they're actually targeting 3 different targets. So the first one will be the CD90, then the second one CTLA4, and the PD1. Okay, so they're using 2 different guide RNA sequence to do the knockout on the targeted gene. Okay, you can see that when different guide RNA sequence is used, they will be different in terms of knockout efficiency. So some region, the guide RNA targeting region, can have a better knockout efficiency. Okay, and the next one will be the thing that show to be crucial will be the Cas9 protein and guide RNA ratio. So therefore, it's important to optimize the ratio between them. Okay, so in their experiment, they're using the ratio of 1:1, 3 different total substrate concentration, and then after that, the ratio of 3:1 and 5:1. And then you can see that in their experiment and their cell type, okay, the 5:1 ratio is actually giving the best KO efficiency. Okay. Okay, so in some cases, guide RNA that you have tried might not work well or give you good enough knockout efficiency. And here in the experiment, they actually provide some unconventional strategy, which is actually use multiple guide RNA targeting the same gene. So they actually show that they are able to get, in some cases, higher KO knockout efficiency when they're using multiple guide RNA targeting the same gene. Okay, so and for this particular one, it will be slightly interesting one because in their publication, Seki, they actually show that there are some influence of knockout efficiency when different commercial Cas9 protein is used. So here they're actually comparing gene art platinum Cas9 and true cut Cas9 protein from commercial available. They're actually from the same company, and one of them actually recently I just found that is actually discontinued. So you can see that the true cut Cas9 actually somehow, even though the sequence is the same, is both also recombinant, both also with nuclear localizing signal. somehow the TrueCut Cas9 actually has a better modification efficiency, which is actually quite interesting. Okay, so as mentioned earlier, the nucleofection of RNP is important for us to actually titrate the optimal ratio between the Cas9 protein and guide RNA. So in most publications, the ratio will be between 1 to 1.2 and 1 to 5. So this could be the range that we can actually optimize to see which one work out the best. And then for the amount, the recommendation when we're using nucleofection, so the concentration will be between 0.08 to 0.8. Okay, yeah. So, and then another thing when we're performing the RNP experiment is whenever we're using for ssODN, the single-stranded oligodeoxynucleotide, for the purpose of targeted modification, maybe we can consider using an enhancer. So there is a commercial available product called ALTR, Cas9 electroporation enhancer. So that actually show to have a better HDR effect when we are using the ssODN. Okay. Another point to take notes will be try to avoid too much dilution of the nucleofection samples by the RNP buffer. Okay, so usually foreign buffer should not exceed more than 10% of the transfection reagent and then consider complexing means that reconstitute the Cas9 protein or guide RNA complex directly in nucleofector solution. Okay, so after transfection, another tips for some post-editing knockout cell screening. Okay, so clonal selection can be started between 24 hours and 7 days post-transfection. So the optimal time point has to be determined depending on the individual experiment setting. Okay, so one option to increase the number of clones is transfecting a vector that co-express a frozen protein. Okay, so this will actually allow enrichment of transfected cell by using flow cytometry cell sorting. Okay, so for cells that do not like to be grown as a single cell, for example, embryonic stem cell or induced pluripotent stem cell, FACS sorting might also be an alternative to the limiting dilution process or using pools instead of a single clone. Okay, so how to actually help for the enrichment of the editing cell? Okay, so and as mentioned earlier, you can use co-transfection of plasmid expressing the gluten protein. Another one is when we are using the ssODN, the single-stranded oligodeoxynucleotide. We actually label it. Okay, so we label the donor DNA, either it's a plasmid or ssODN, with, let's say in this particular case, Alexa 647. Okay, then after that, we use the flow cytometry cell sorting to sort for cell actually with this particular dye. Then it actually helps to enrich genome editing cell. So in this, it actually show that it can increase the enrichment of this particular cell. Okay, so higher amounts of Alexa 647 labeled donor DNA can increase the HDR frequency after sorting. Okay, so another strategy to actually enrich HDR or NHEJ edited cell is to use a core editing strategy. So this particular strategy is actually transfer another guide RNA and just another guide RNA that editing target called ATP1A1 locus. Okay, when we actually call editing this particular locus, the edited cell will actually become resistant to Ourbain. Okay, so ATP1A1 is an ubiquitous and essential ion pumps, and Ourbain is actually an inhibitor of this particular pump. Okay, so if let's say the cell are not edited, there will be a cell death in between within 48 hours. So therefore, the strategy is to introduce a gain of function modification at ATP-A1. Okay, so via HDR and SSODN. So edited cell will get resistant to Ourbain treatment. So when we're doing core editing, so the cell that actually edited for ATP-A1 will most likely contain the addition, the modification that we want to change as well. Okay, so this particular strategy has been tested when using the K569 cell line and also the CD34 cell. Okay, so in summary for today sharing, okay, so nucleofection offer efficient non-viral transfection of many cell types, including primary cells, stem cells, and cell lines. The advantage of nucleofection is to offer substrate flexibility and simple core transfection due to same condition for various substrates. Okay, so it's suited for DNA, mRNA, or RNP mediated CRISPR genome editing. It's a proven technology with a monthly growing list of genome editing publication and used by many renowned players in the field. So the nucleofection can be easily adapted to different CRISPR approach. So even if you're using CRISPR-A or CRISPR-I or other genome editing system like ZFN or Talent. Okay. So due to the availability of different platform of nucleofection, it can be used for higher throughput screening experiment, as well as for large scale application. Okay, with that, I shall end my sharing today. Thank you for your attention. Okay, so we shall move to the Q&A session if you have any question.
Yeah, thank you, Chee Hong, for the sharing. So, do you guys have any question for our speaker today? Yeah, we have question from Selina Tan. May I know the guide RNA we need to design ourselves? If yes, may I know what should we consider when we design?
May I know, can you repeat the last part of the question, Minty?
The last part of the question is, if yes, may I know what should we consider when we design?
Oh, okay. So, Selina, thanks for your question. Yes, the RNA has to be designed by you, or in some cases for mammals, for human cell, actually you can try to look for a group of scientists called Zhang Feng, ZF. Okay, so they are from MIT. They actually design guide RNA sequence targeting many human genes, and you can look for it from there. And then during the design, actually they are designing tools, so therefore you just need to use the designing tools. So to name a few, there are several designing tools, and you can actually find it from NEB website. So maybe I do a search later, then I will share with you with the link about the guide RNA designing tools available in the online. So when during designing, most of the parameter will be actually set by the designing tools. So therefore, no worry. But the only thing that I would like to add on will be maybe you want to consider choosing more than one design sequence, because sometimes, as shared earlier, the sequence, the region that we actually targeted might have different effect. Some actually works better than others. Therefore, sometimes we will perform something called a mutation efficiency test to check which one actually has the best efficiency before we actually continue with the experiment. Because actually, to me, the experiment is actually quite straightforward. But the harder part might be the screening of the editing cell. So before we actually screen for the editing cell, so maybe we want to test for which guide RNA actually has the best knockout efficiency, then only we proceed to the screening to look for the cell in the case that you want to look for the edited cell. Yeah.
All right, thank you, Chee Hong. So we have another question from Ahmad Basli Ramzi. Hi, many thanks for such an insightful talk. Could you share if the RNP method has also been used for Cas12 protein?
Actually, I'm not really familiar with the Cas12 protein, but if I'm not mistaken, it should work quite similarly to the Cas9, because it's actually only with sequence difference and also the PEM sequence different. So therefore, if you're using the RNP method, it should work quite similarly. Yeah. So another thing I would like to highlight when using different Cas protein, so we have different what we call a PEM sequence. So PEM sequence is actually short form for PAM. So unfortunately, when we are using the Cas protein, we have one restriction, which is actually the targeting site has to be followed with a PEM sequence. For example, Cas9, we need to follow with NGG. Okay, N stand for ATCG, so it's either AGG, TGG, CGG, or GGG. So for Cas12, then they have a different PEM sequence. So when you are but however, in the designing software, you automatically should look for this particular PEM sequence. Next.
Yeah, we have another question from Masani. Nice talk. Are there any works or literature using Nucleofector for plants?
Oh, okay. So thanks for your question. So unfortunately, Nucleofection currently only works for mammalian cell. So if you are working for plant, maybe you can contact us separately. But mostly for plant, as far as I know, you the how to say, the delivery of the guide RNA and Cas protein, as far as I know, is actually using the standard transformation approach, which is the Ourbain. So perhaps we have to look for the tools for that delivery method. And luckily, we have the Plasmid curator site. I'm not sure you guys know about it, which is actually the AdGenes, which is proved to be a valuable tools for the scientific community. So a lot of science, researcher, they're actually depositing their plasmid DNA used for various applications, even for CRISPR, actually depositing their CRISPR plasmid tools in the AdGene site. So therefore, you can search in the AdGene site looking for, let's say, plant CRISPR editing, and then after that to look for the plasmid for the plant editing purpose. For the delivery method, then if I'm not mistaken, they're using the agrobacterium method. So you're just using the suitable plasmid and then actually put into the agrobacterium and then use it to infect the plant. And hopefully the agrobacterium can infect your particular plant organism.
Thank you, Chee Hong. Do you guys have any more questions for our speaker today? So if there is no further question, we shall end our webinar session today. So thank you guys again for joining our webinar session today. And if you have any question regarding the CRISPR transfection using Nucleofector technology, feel free to contact us at sales@nextgene.my, and we will send the recorded video of this webinar, the slides, and also to you after the webinar session. All right, thank you and have a nice day.
Thank you everyone for joining. Stay safe.